Okay, so welcome to this next video in which we are discussing the HIV life cycle and also antiretroviral drugs. Right, so those of you who have watched the previous video will know that I have suddenly jumped onto a new clean sheet of paper and that the other video stopped rather abruptly. The reason for that is that I started making an embarrassingly awful mistake with the Latin alphabet. So we've got a new piece of paper now where the mess isn't present anymore, and we're going to start again. So the different clades of HIV we were talking about. So we were talking about the fact that HIV-1 could be further divided into these 4 separate groups. Okay, so we've got the M group, the N group, the O group, and also the P group, which came around in 2009. Okay, we then discussed that the M group of all of these was by far the most important. Okay, and this contains 9 different clades, okay, which are all genetically distinct strains. Okay, and the different clades are clade A, clade B, clade C, clade D. There is no clade E. It then goes to clade F, clade G, clade H, okay, then clade J, and then clade K. Okay, so it doesn't follow the alphabet perfectly. Okay, so there are 9 distinct clades here, which are all within the group of M, basically, of HIV-1. Okay, now, of all of these different clades, the most important clade is MB. Okay, so HIV-1, Group M, Clade B, is the most important form of HIV, basically. It's the form that is really, really common within the US and Western Europe. Okay, so it's one of the major forms that infects humans worldwide. Okay, right. So, now let's move past this classification and let's go on to talk about the actual infection of cells by this virus. Okay, so basically, the first thing we need to talk about is the fact that this virus is going to bind to a protein called CD4. Okay, so let's draw our little virus particle. Now we'll have a chance to revise our virion structure. Okay, so here is our viral envelope. Then underneath the viral envelope we have the P17 matrix protein here. Okay, within the P17 matrix protein we then have the nucleocapsid, which is this cone-shaped capsid made out of P24, and then within the capsid you then have the 2 pieces of single-stranded positive sense RNA, and then you have these 3 important enzymes, reverse transcriptase, integrase, and protease. Okay, so here is our HIV virion particle, and then of course we know that on the viral envelope you have GP41 proteins which are attached to GP120 proteins. Now basically the infection cycle begins with GP120 here, and I think since it's one of the stars in this phase of the video it should be coloured in. So we'll colour GP120 in pink here. Okay, and it's attached to the GP41, which then is attached to the membrane. Okay, so we'll colour in GP41 in blue here. There we go. Right, so the infection is going to begin with GP120 binding to CD4 protein molecules on the surface of human cells. And I should just say that I'm not going to go through the actual pathology of the, of becoming infected, of the whole human becoming infected. I'm going to focus in on the viral life cycle, the infection of an individual cell, rather than the infection of the whole human, basically. I mean, everyone knows how you become infected with HIV. Generally, anyway. Basically, the way that it gains access to cells is through the GP120 binding to a protein on the surface of the cell. Okay, and this confers the specificity of the virus for certain cells, and this is why it specifically attacks the immune system, because this protein that is going to be on the surface of the cell, and I'll draw the phospholipid bilayer of our cell here. Okay, so this is the phospholipid bilayer. Okay, this protein is cluster of designation 4, CD4. So this is short for cluster of designation or cluster of differentiation 4. So the C is for cluster and the D is for designation or differentiation. Designation 4. Right, so let's colour in CD4 in orange. Now, which cells actually have cluster of designation 4 proteins on their surface? Well, basically, they're immune cells. Okay, so there is a huge group of T lymphocytes which have CD4 on their surface, and these are called CD4 positive T cells. Okay, so T cells as a population can be divided into two major groups. So there are CD4 positive T cells, and then there are CD8 positive T cells. And a T cell either has CD4 on its surface or it has CD8 on its surface. Okay, so a huge population of T cells are CD4 positive and do have CD4 on their surface, and these are going to be the targets for HIV. Okay, and these are a major part of the adaptive immune response. You cannot start an adaptive immune response without these guys, so they are extremely important. Okay, right. Maybe you can start an adaptive immune response without these guys. The cell-mediated response through the CD8s can be started, but it will be very weak without the CD4 positive T cells. Okay, so let's now talk about other cells that have CD4 on their surface. So monocytes also have CD4 on their surface, and monocytes are a type of immune cell that circulates within the bloodstream. Okay, and basically when they are recruited to a site of inflammation, okay, so they move from the bloodstream into the interstitial fluid across the endothelium, okay, they will then differentiate into one of two types of cells, and they can actually differentiate into more than just these two types of cells, but these are the main two types of cells that they differentiate into. They differentiate into macrophages. So basically the monocyte is the precursor to the macrophage. You don't have macrophages circulating within your bloodstream. You have monocytes which then differentiate into macrophages when they get to the site of infection. Now the shorthand for macrophages, just so that you know, is to put M and then phi, the Greek symbol phi, like so. That's a shorthand for macrophages. The other key cells that monocytes can differentiate into is dendritic cells. Okay, and dendritic cells have a similar function to macrophages. They phagocytose pathogens and they present antigens on their surface in MHC class 2 along with co-stimulatory molecules, and they can activate CD4 positive naive T cells. They can activate the adaptive immune response, and they're very, very good at doing that. Now, both macrophages and dendritic cells, which are derived from monocytes, also have CD4 on their surface, so these are going to be taken out then by HIV. They're going to be targets for HIV. So these three types of cells, CD4 positive T cells, macrophages and dendritic cells, and also obviously monocytes within the blood, these are all going to be targets for the HIV virus, okay, because of the presence of CD4 on their surface. Right, now CD4 on its own is not enough for the virus to be able to get in. There's also a co-receptor. So basically what happens is initially the GP120 binds to the CD4, okay, that triggers a conformational change in the GP120, and the GP120 now gets a new binding site where it can bind to another protein on the surface, and this is what's known as the co-receptor for GP120, and to get in it now has to bind to this co-receptor. Okay, so these co-receptors are going to be chemokine receptors. So what I now want to talk about is I want to talk about what chemokines are and what the different receptors for chemokines are, and then I want to look at the specific chemokine receptors which are targets for GP120, because there are two of them, and basically depending on how long you've had the infection will determine whether the HIV uses one or the other generally. Okay, so let's now talk about chemokines then. So chemokines is the next topic. Okay, so that we can understand what a chemokine receptor is. Okay, and this is just going to be a little brief topic. So basically, chemokines are proteins that are important in the immune response. Basically, chemokine stands for chemoattractant cytokine. Okay, so chemoattractive cytokine. So the chemos for chemoattractive, and then the kines is for cytokines. So many of the molecules that are involved in the acute inflammatory response are called cytokines. They're basically molecules which signal between cells and trigger inflammatory activity. Okay, so chemokines all have a conserved structure basically. So let me now show you the structure of these proteins because it's quite nice. Okay, so you start off with the amino terminus here, and basically what happens is the polypeptide comes round like so. Okay, and then it loops behind itself, so it's now going to go behind itself here. So this is now all behind what we've got here. So you can imagine this all being in one plane, and now this is behind the piece of paper, if you like. So we're going into the page further back, and then what we have is a special beta pleated sheet here. So we have 3 beta strands, which are all in this beta pleated sheet, and then the protein comes back forward, and then we'll go in front of the beta pleated sheet and finish off over here. Okay, so right, let me highlight special portions of this structure. So basically, this portion that you have in front of the beta pleated sheet, and we can imagine that it's in the same plane as the piece of paper basically, which is in turquoise here. Okay, this is an alpha helix. That's why I'm highlighting it as turquoise. It's an alpha helical structure. Okay, we've then got these 3 beta strands here which form a little beta pleated sheet. Okay, like so, and this is behind the page. This is in a plane behind the page. Okay, and then this final portion here is in front of the page. Well, sorry, is within the page. So you can imagine that the turquoise alpha helix and this portion here are within the page, and then the beta pleated sheets are behind the page. Okay, so these are beta strands in orange. Okay, and basically we now want to talk about why this thing is held together. What holds this thing in this shape? Well, basically there are some really important cysteine amino acids. So basically you have 2 cysteine residues that are really close to the amino terminus. So you're going to have 2 cysteine residues here, and I'll show you the structure of cysteine residue in a moment. So I'll highlight them in a special colour. So these in pink here, these are cysteine residues. Okay, and we'll call this cysteine number 1 here, cysteine number 2 here. Then you also have a cysteine residue over here. That will be cysteine number 3. Okay, so if you follow the polypeptide round, this is the next cysteine. Okay, and then continue following the polypeptide around again all the way to here. The final cysteine is then going to be cysteine number 4, and it's at that position there. And basically, what's going to happen is that cysteine number 1 is going to bind to cysteine number 3, and cysteine number 2 is going to bind to cysteine number 4, and that's what holds this structure together. So let me show you the structure of cysteine amino acid, and then I'll show you the bonds that 2 cysteine amino acids are going to form, and how that's going to help hold the whole structure together. So the structure of a cysteine amino acid then, and I'll show this as though we're looking at the cysteine residue, i.e. the cysteine amino acid as though it's within the protein, basically. So here's the amino group, which obviously faces this direction up here towards the amino terminus. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, which again will be linked to an amino acid behind it. So I'm drawing it as though it's within a polypeptide. We can imagine there's amino acid here, and there's another amino acid here. Okay, now the R group of the cysteine amino acid consists of a methylene group with a thiol group coming off it, like so. Okay, so Basically, when you have a sulfur atom with a hydrogen atom coming off, that's called a thiol group, and its chemical properties are very similar to alcohol groups because sulfur is in the same group at the periodic table as oxygen and therefore has similar properties to oxygen. And of course, if this was an oxygen here, that would just be an alcohol group, and therefore the thiol groups have similar chemical properties to alcohol groups. Okay, so we have these cysteine amino acids at position 1, 2, 3, and 4 within our chemokine. Okay, and what can happen is these cysteines can bind together, and let me show you this. So I'll show you it as though we're talking about cysteine 1 and cysteine 3. So let me draw another cysteine, some more cysteines out. So here again is cysteine 1 here. So there's the amino group, the alpha carbon, the carboxylic acid group, and then the R group now is going to be a methylene group with the thiol group, but I'm going to be fortuitous and not draw the hydrogen coming off here, because in fact they're going to form a disulfide bond, and the hydrogens are going to, they're not going to be there anymore. Okay, so let's now draw the cysteine residue at position 3 here. So amino terminus, let me just figure out the direction the amino terminus is going to be in. So this is the amino side of cysteine 3, and this is the carboxylic acid side. So again, it will be like so. So I have the amino group up here, and I don't know if I've left enough space. Then we'll have the alpha carbon. I'm going to have to squash in the R group here. Here's the methylene group. And now here's the sulfur atom, and basically they're going to bind together like so. Here's the hydrogen off the alpha carbon, and here's the carboxylic acid group, which will be bound to the next amino acid along. Okay, so this link between these two sulfur atoms here, this is the disulfide bond, okay, or a disulfide bridge. So you've got these disulfide bonds between these cysteine residues within the chemokine structure. One between cysteine 1 and cysteine 3, and one between cysteine 2 and cysteine 4. And these bonds are very, very strong, and they hold the whole chemokine structure together. So all chemokines have this same conserved structure, basically. This is what characterizes chemokines. Now, chemokines can then be split into 4 groups. Okay, so the first group, the major group, is the CC chemokine family. Okay, so there's the family of CC chemokines, and now what we will do is explain the notation basically. So why is this family called the CC chemokines? Well, basically, in this family, all of these chemokines have cysteine 1 and cysteine 2 actually next to each other. Okay, so the naming system for the chemokines, the different families of chemokines, basically what it tells you is the positions of cysteine 1 and cysteine 2 relative to one another. And basically, in the CC chemokine family of chemokines, the two cysteines, cysteine 1 and cysteine 2, are absolutely next to each other. So you have cysteine 1, okay, so if I draw this structure out again, you'll have the amino group here, okay, and then at some point you have cysteine 1, and then immediately following it you have cysteine 2, and C is the single letter amino acid code for cysteine, which is why I'm using it there. Okay, then the rest of the chemokine structure follows, so it loops around. You have these 3 beta-pleated sheets, and then the alpha helix ending with the carboxylic acid group here. And then you have cysteine 3 here, which will be disulfide linked to that. And then you have cysteine 4 here, which will be disulfide linked to cysteine 2. Okay, so all CC chemokines have these cysteine 1 and cysteine 2 absolutely next to each other, i.e. there are no amino acids in between them. Okay, so you might now be able to guess the reasoning behind the namings of the other families. Okay, so next family is CXC chemokines, okay, and the CC chemokines and the CXC chemokines are the main 2 families of chemokines. The other 2 are kind of minor. Okay, so basically in CXC chemokines you have 1 amino acid in between the 2 cysteines. So in between the first and second cysteines of the chemokine structure, you have a single amino acid, and that's this amino acid denoted by X. Okay, so instead of them being right next to each other, you have 1 amino acid in between them. Okay, next family is CX3C. chemokine family. Okay, and again, this makes complete sense. This time, you've just got 3 amino acids in between the 2 cysteines. So in between the first and the second cysteine, you now have 3 separate amino acids, basically. So you have to fit in 1, 2, 3 amino acids before you then go to the final cysteine, the second cysteine. Okay, so all of those 3 are very, very simple. There is one that is a little bit more complicated. Okay, and the final family is a little bit more complicated. So the final family is called the C chemokines, or sometimes it's called the XC chemokines. Okay, and this one I'll have to draw out another picture. So basically, in XC chemokines, you don't have cysteine 1 and you don't have cysteine 3. So that's why this X is here. It basically tells you that there is no cysteine 1, basically. So you only have one disulfide bond holding the entire thing together in XC chemokines. So here is the amino group. It'll go around like so. You'll then have the 3 beta pleated sheet. Well, you'll have the 3 beta strands which form a beta pleated sheet, and then the carboxylic acid group here. And basically there's no cysteine 1, but there is a cysteine 2 here, and then that will be linked to cysteine 4, which will be here. Okay, so there's no bond between 1 and 3 because 1 is not present. Okay, so those are the XC chemokines, and the X shows you that there is no C1, basically. There's no cysteine 1. Okay, but as I say, the main 2 families are the CC chemokines and the CXC chemokines. The other 2 are kind of niche, basically. CX3C chemokines and C chemokines are much more niche. Okay, right. So let's now talk about chemokine receptors. So basically, chemokine receptors usually come in the family of rhodopsin G protein coupled receptors. Okay, so the 2 chemokine receptors that we are specifically going to be interested in are CCR5 and also CXCR4. Okay, so what do these stand for? Well, this stands for CC chemokine, that's what the CC stands for, and then receptor 5. So this is a receptor for CC chemokines, and we now know what that means. Okay, and CXCR4 stands for CXC chemokine, and again we know that that's a massive great family of chemokines, and then it's receptor 4. Okay, right, so I want to talk about what the general structure of these chemokine receptors are, and it goes really across the entire board of chemokine receptors, but it certainly covers these 2. Basically, they are G protein coupled receptors. Okay, so G protein coupled receptors, and basically the family of G protein coupled receptors is enormous. There are over 800 different G protein coupled receptors that are known about. Okay, and to help us make sense of the different G protein coupled receptors are usually grouped into one of 5 families. Okay, so the biggest by far family is the rhodopsin family of G protein coupled receptors, and these are the G protein coupled receptors that are similar in structure to rhodopsin, the retinal pigment. Well, the retinal photoreceptor. Okay, so basically over 750 of the known G protein coupled receptors are in this family. So the other families are much more niche, basically. But there are other families. There's also the secretin family of G protein coupled receptors, the glutamate family of G protein coupled receptors, which contains the metabotropic glutamate receptors and the GABA B receptors. The secretin family contains things like parathyroid hormone receptors, glucagon receptors. Then we've also got the adhesion family of G protein coupled receptors, which contains a whole bunch of G protein coupled receptors which bind to extracellular matrix. And then finally, you also have what's known as the frizzled taste 2 receptors family. Okay, and this is named after the two main members that are within it, which are the frizzled receptor, which is a receptor for the ligand Wnt, and the taste2 receptor, which is a receptor for bitter compounds and is used in the perception of taste. Okay, but the main family of G protein coupled receptors is this rhodopsin family. So I'm just putting those there just to make the discussion complete. But the chemokine receptors are all within this rhodopsin family of G protein coupled receptors. Now let me explain what that means. Basically, rhodopsin G protein coupled receptors are very simple structure. Basically, they have a very small amino terminal domain that's extracellular. Then they have these 7 membrane-spanning alpha helices, like so, so the typical structure for a G protein coupled receptor. And then they have a small carboxylic acid terminus as well. And the characteristic feature of members of the rhodopsin family of G protein coupled receptors is that they bind their ligand through their transmembrane domains, basically. So the ligand will bind to the G protein coupled receptor by interacting with residues that are in the transmembrane domains. Okay, so here is the ligand, basically. So that's the characteristic feature of all of these rhodopsin family G protein coupled receptors, that the ligand binds to residues which are actually within these 7 membrane spanning alpha helices. And I've drawn it obviously on transmembrane domain 3 and transmembrane domain 4, but that's just, it's just a picture. It could be binding to residues on other transmembrane domains as well. The important thing to understand is that it binds to residues within the membrane spanning alpha helices. Okay, so we'll call it there for this video and continue our discussion of the HIV life cycle in the next video.